All right, so these are your notes for section 3 of chapter 8, biology. This section is on DNA replication. So our objectives are to summarize the process of DNA replication and then also describe the role of enzymes in DNA replication. Okay, so first off, replication, remember, that's the process through which DNA is copied during the cell cycle. If you guys remember, if I was here with you, I'd probably make this a whiteboard question, but during the cell cycle, that DNA is replicated in that S phase, right? It's replicated during S, which was short for synthesis. And this is super, super easy, guys. Right? Replicating DNA works because it's really, really simple. All you have to do is use those base pairs that we talked about. If you know, again, that A always goes with T and that G always goes with C, you can do almost anything on this next test. So all you have to do is look at this strand of DNA that I have down here and match it up with base pairs. So if I have a T to begin this, that T should go with an A. The A goes with the T, the G goes with the C goes with the G, and so on and so forth. So you can copy as I fill this thing out, but really it's a lot easier for you just to do it yourself. So make sure that you are using your base pairs, but you can just fill this in the rest of the way. And you might make a mistake every once in a while, but you're much more likely to make a mistake if you're copying. Right? If you do it yourself and you use those base pairs, you can do it really quickly, really easily. My new strand of DNA is complementary to that initial strand of DNA. All I had to do was match up A with T, G with C. So that's essentially how DNA is replicated. Since A always goes with T, to bring in a new nucleotide to build a new strand, all I have to do is base pair. So it's a really simple process that we're going to talk about in a complicated fashion, but just keep in mind that all you really need to know at the end of the day is that A goes with T, G goes with C. The rest of the stuff is important, but being able to do it and matching up those base pairs is probably more important than all the other stuff. So don't make it more complicated than it needs to be. Okay, so we are going to talk about the replication process, which involves some enzymes, and it's somewhat complicated, but again, at the end of the day, all you're doing is base pairing. A goes with T, G goes with C. So don't make it too hard. The first step, DNA helicase unzips the double helix. It starts somewhere in the middle and goes both directions. So you gotta imagine you got this piece of DNA. Remember, it's that double helix structure. And before I can start copying one side or the other, I have to open up that DNA. I have to separate those two strands to do what I need to do. So I got a piece of DNA here and I've opened it up. I've separated those two strands so that they're no longer wound around each other in that double helix. And that's the first step in copying your DNA. You have to separate those two strands. Once those two strands are separated, you have bases that are exposed. Right? Within this DNA, right, I'm just going to draw in a random sequence here, right, there's a code. You have random base pairs on both sides. But if it's AGTTCGCAATCC, like I have drawn above, down below should be complementary to that. So an A goes with the T, a G goes with the C, so on and so forth. My other strand would look something like this. Right, so the strands are complementary again. You got base pairs, but we're talking about replicating this DNA. So I already have this piece of DNA with these complementary bases. In order to replicate, I just need to bring in new nucleotides and attach them to the old ones, pair them up with the old ones to make this work. Now remember, we're trying to talk about the complicated process, mentioning some of those enzymes that help along the way. This DNA helicase is an enzyme that essentially works kind of like a zipper does on your coat, or your jacket, your pants, whatever, right? As it moves along the DNA, it unzips that DNA, right? So you're just pulling it down the zipper and the two strands are separating from each other. So I got this DNA helicase that's helping me unwind that DNA. It's exposing those two sides, separating those two sides so I can start bringing in new nucleotides. The next enzyme that I'm worried about is called DNA polymerase. DNA polymerase is actually going to build that DNA polymer. It's going to start bringing in new nucleotides to build those two strands that I need to add in here. So one of the next things that we need to worry about here is there's always a directionality. We're going to put labels on this DNA, and we kind of have to know what ends what. So if I work my way backwards from this top strand, right, through my little wind over here, this is going to be my 3' end. I'd follow it back. This is 5'. Ooh. Pen was being weird there. So this is the 5' end. And if this is the 5' end up here, then this is going to be a 3' end here. And work my way backwards, this must be a 5' end over here. So we always have this idea of anti-parallel. If you go back a couple slides here, with this 3', there should have been a 5', and on the other end, with this 5', there should be a 3'. It's always anti-parallel. If one strand is running in one direction, the other one's going to be going in the other direction. So I just need those labels to indicate what direction I'm going, or which way each strand is facing. Polymerase, though, always builds from 5' to 3'. And that's going to cause a problem when I am replicating this DNA. Because as of right now, I have a little bit of this DNA opened, and I can start bringing in new nucleotides using that DNA polymerase. Right? It can start right in here, start moving along this strand, and attract nucleotides to go with those current bases that are already there. So this A goes with the T, this G goes with the C, so on and so forth. And it can keep going down this strand. To build my new piece of DNA in this direction, because if this is 5', this is going to be, 3'. But that's the only direction that DNA polymerase can build. My other strand then is going to be built in the opposite direction. So if I'm building this strand on top going from left to right, the bottom strand is going to have to be built the other direction, because it's going to start over here with 5', and you need to build towards a 3' on the other end. So I'm going to build this way as I'm bringing in bases on this side. So I can have another DNA polymerase come in here and I can start building, adding in nucleotides, using base pairs. But I'm going to be building in opposite directions off of either strand, which leads to a problem as we go on. Okay, so since DNA polymerase only builds in one direction, that means that I'm going to have leading and lagging strands being built. And ultimately, that means that I'm going to end up with some fragments that need to be linked by DNA ligase. Now, for now, we're probably just going to leave it as is, but we'll come back later and talk about those fragments and talk about the leading and lagging strands. We'll just have to review that on Monday. Okay, but at the end of the day, your end result, you have two identical DNA molecules. It's going to be semi-conservative because you have one new to go with the old. Right, the semi-conservative part of this, right, you have an old piece of DNA that's black to go with your new piece of DNA that's orange. Right, you're always going to have a little bit of old with new. So that word, semi-conservative, is just telling you for every new piece of DNA, you had an old template that you worked off of to build it. All right? Okay, so as far as those enzymes are concerned that we talked about, whether it's helicase or polymerase or whatever, right, you can always recognize that it's an enzyme because it always has that same suffix. It always ends with this ase, right? Whether it's polymerase, helicase, ligase that we'll talk about later, right? So you can always tell that it's an enzyme because you see this ase at the end of it. But you can usually tell what an enzyme does by its name too. When I say DNA polymerase, and you can see DNA polymer in the name, that's probably what it's going to do. It's going to try and build that DNA polymer. Right? That's its job. So usually you can tell what an enzyme does by its name. So just as we go through and you see those different words, just make sure that you recognize that their jobs are fairly apparent most of the time. The name, what it does is usually in the name. So DNA polymerase, you build that DNA polymer. DNA ligase, it's going to link two different pieces of DNA together. Helicase unwinds that helix, right? So you got key words that you can look for, key parts of the name that you can use to tell you what it does. Replication is remarkably fast and accurate. DNA polymerase can actually add about 50 nucleotides per second, and it's going to start at many different origins at the same time. So you're not just going to have one replication bubble within a piece of DNA, you're going to have multiple replication bubbles. So it might be open here, also open elsewhere, right? It's hard to draw this in the limited space that I have here. But if you have this piece of DNA, I didn't just have to open it right here with that DNA helicase. I'm also working the other direction, opening it that way, but I could also open a bubble right over here if I wanted to, right? So you're going to open lots of little bubbles and you're going to start replicating in lots of different places. Remember, direction matters. So I'm going to be building one way, building the other, depending on which side's 5', 3'. But you can do this process really, really quickly. So your cells can replicate your DNA really, really fast, and it's really, really accurate. So we use multiple origins Prokaryotes only start at one point, but that's easy for them because their DNA is really short. It's circular. So they start at one place and work their way around and it's done. So it's pretty quick, pretty easy for them. The other thing down here, that polymerase is going to proofread for you, which is pretty important. Only about 1 in a billion mistakes doesn't get fixed. So every once in a while, you'll have those base pairs going, and you'll have an A that goes with a G by accident, right? So you got this little strand going here, and every once in a while, you'll have an error in there. So you put a C with a C instead of a G with that C. But polymerase will proofread that. It'll see this error, it'll fix it, and replace that C with a G. And only 1 in a billion times does it not see it, does it not fix it. So your DNA is replicated really fast, but it's also really accurate. It's almost always done exactly the way it's supposed to be done. If you don't do it accurately, you could end up with mutations. You could end up with problems. And we'll talk about those later. But generally speaking, your DNA polymerase does a really good job of that. It does a better job than you would. Right? If you start, as we go through this chapter, and you start replicating or you start reading DNA, you're going to make more mistakes within this chapter than your DNA polymerase has made in your lifetime. So it's really, really good at what it does. It always brings in that right base. And again, that's pretty important because your DNA has actually been replicated trillions of times since you were a single cell. Right? In order for you to grow into the organism that you are, your DNA had to be replicated over and over again. So it's important that it's done quickly, and it's important that it's done accurately so that all of your cells function the way that they're supposed to function. All right, so other than these notes, you guys have a packet already that I gave you for Chapter 8. You just need to start working on your Section 3 worksheet. And again, we're going to come back and touch on some of this stuff again on Monday. But for right now, we're good. So I can turn off this recorder.